Welcome on this ECHA webinar on new developments and regulatory applications of the OECD QSAR Toolbox. My name is Andrea Gissi and I am a scientific officer at the European Chemicals Agency. What you can expect today are demonstrations and practical applications of the QSAR Toolbox. We will see recent and future developments of the software, including the work supported by the US Environmental Protection Agency. We will also see how to use the QSAR Toolbox for regulatory applications, with a focus on read across for REACH. Only a few months ago, we had another ECHA webinar on the OECD QSAR Toolbox. This one is different because it will focus on practical applications of the software, as you requested in your feedback from the previous webinar. We will also have a live question and answer session. To ask your questions, connect at the website slido.com with the event code Qsr Toolbox 22, or just use the QR code on the slide. You can send questions between 2 p.m. and 4:30 Helsinki time, and only questions within the scope of this webinar will be answered. If you see that your question is not answered, please contact us via the official website. In the live panel, there will be 4 of us from ECHA. There will be me as QSAR Toolbox project manager, then Doris Hierman, that is a QSAR expert, but also the chair of the PBT expert group, Andrea Richards, who is an expert of read across and dossier evaluation, and Tomasz Sobański, who works on new approach methodologies and QSR assessment. The material from this webinar will be made available as video recording. We will also have the presentations, the question and answer that will be published soon after the event, and also supplementary video where you will find even more demonstrations about the QSAR Toolbox, for which there was no time in the main agenda of the webinar. Here we can see the program. After my welcome, there will be the presentation More Than QSAR Predictions. Here, Patience Brown from OECD will talk about the functionalities in the toolbox. And then following that, Doris Hierman from ECHA will demonstrate how to use those functionalities in practice on the QSAR Toolbox. The next presentation is from the contractor developing the QSAR Toolbox, the Laboratory of Mathematical Chemistry of Burgas University, LMC. And from them, Darina Jordanova and Stanislav Temelkov will present what is new in Toolbox version 4.5, with a focus on the automated workflow for defined approaches for skin sensitization and on the web client. Following that, there will be a presentation by the US Environmental Protection Agency, EPA, from Martin Phillips and Brian Racour. They will present QSAR Toolbox at the US EPA. Following that, I will give you a presentation about the link between QSAR Toolbox and Euclid. After me, Andrea Richards will give a presentation on QSAR Toolbox in support of read across for REACH. After this presentation, there will be a new functionality that we didn't have in the previous Toolbox webinar, that is a live Q&A panel, not only in writing in Slido, but we will answer orally your questions. And this will be a mix of the questions that you have sent in advance when you pre-registered for the webinar. We got many pre-registrations, many questions. Thank you for that. But we will also answer some of the questions that you will send during the event in Slido. And finally, Tiago Pedrosa, the new head of Computational Assessment Unit, will provide some conclusions with a view on the future developments on the toolbox. If you need more resources on the software, visit the Qsr Toolbox website, where you can download for free the software, but you can also find tutorials, manuals, and other material. Then you can watch the recording of the webinar we published last year, OECD QSAR Toolbox applications for REACH and beyond, if you are curious to see how regulatory authorities around the globe use the QSAR Toolbox. Finally, you can visit the QSAR Toolbox page on the OECD website. And after this introduction, it's now time to start with the webinar with the presentation, More Than QSAR Predictions.
Good day to everyone and welcome to the webinar on the OECD QSAR Toolbox. My name is Patience Brown and I'm with the OECD Environmental Directorate. And it is my pleasure today to give an overview on the QSAR Toolbox and how it is more than just QSAR prediction. So first, I'd like to just give a brief description of the governance of the Toolbox. ECHA and OECD co-own and co-develop the Toolbox, and this is really more than a decade of collaboration on this particular project. The work is executed by the developer, that's LMC, the Laboratory of Mathematical Chemistry, and these activities fall under the OECD umbrella, specifically the overview of the Working Party on Hazard Assessments, and the core development is contracted by ECHA. So there's a coordination group that involves the OECD Secretariat, members of ECHA, and the IT developers. And in addition, we receive input from the Toolbox Management Group, which is an OECD group including not only the members of the coordination group, but also nominated experts from industry, regulatory authorities, and non-government organizations. In addition to this very collaborative input, we also have partners that develop their particular products in the context of the OECD QSAR Toolbox. For example, U.S. Environmental Protection Agency has supported linking the toolbox with their tool Oncologic, which predicts the carcinogenic potential of chemicals. And Japan developed a plugin for their QSAR Toolbox, Kate. And you'll hear more about the Oncologic connection with the QSAR Toolbox later in the webinar. In addition to these specifically supported projects, there are the potential of third parties to make their own extensions available through the Toolbox repository of tools. So the QSAR Toolbox is used globally by regulators. We can see members that are called out in North America, South America, Europe, Australia, and Asia. But in addition to the regulatory authorities, the Toolbox is used by members of industry and academia to fill data gaps. And I'd like to talk a little bit about how exactly that's done. So as you saw in the title of this presentation, the QSAR Toolbox is more than just QSAR predictions. It is in fact a free software application that supports toxicologists and ecotoxicologists in performing reproducible and transparent chemical hazard assessment using non-animal data. So there are a variety of ways that this can be done. Users can search for specific chemicals using CAS or chemical name or structure, as well as groups of chemicals by databases or inventories, for example. And once input into the QSAR Toolbox, users can retrieve experimental in vitro and in vivo data, as well as profiling chemicals by physical chemical properties. They can simulate metabolism from the parent compounds. They can identify analogs and build categories. And they can predict the effects of chemicals or groups of chemicals. And all of this is included in the report generated by the QSAR Toolbox. So this really helps to bridge the need for de novo animal testing for chemicals for which data are not currently available. In addition, in some cases, the data may actually be available in publicly searchable websites and databases. For example, experimental data is easily accessible through the QSAR Toolbox, and in many cases, there may be no need to perform a study if there's already information. The Toolbox searches experimental data on chemicals from almost 60 databases, and included in that is 100,000 chemicals and 3 million data points. So it's really quite a bit of information. This includes databases, for example, such as the REACH chemicals database, the European Food Safety Authority Food Tox database, and the U.S. Environmental Protection Agency's ECOTOX database, among others. And you'll hear more about the link between the QSAR Toolbox and the IUCLID database later in this webinar as well. So experimental data from analogs can also be used for read-across predictions, and we'll talk a little bit about how that can be done. So I mentioned the various profilers available in the Qsr Toolbox. There are structural profilers that support identification of structurally similar substances, but in addition, there are mechanistic profilers that provide information based on an understanding of the mode of action, which is key to predict the activity of substances or to form mechanistically sound categories. The results of the profilers in the toolbox include descriptions and references of the scientific papers to explain the outcome, and these can be linked through the report and accessed by users. So, of course, metabolism influences the activity of substances in many cases, and it's important to take into account when making predictions or forming categories. One of the key identified limitations of many non-animal methods is the limited ability to predict the effects of metabolites when using non-animal methods to simulate animal data. So the toolbox adds key functionality in this regard. Results from 5 experimental metabolic databases and 11 metabolic simulators can be taken into account when assessing a single substance or groups of substances or categories. So how is all of this information used? The toolbox supports predictions based on analogs and categories, and it can take into account the profilers I mentioned, the structural and mechanistic information, as well as the metabolic aspects to find toxicologically relevant analogs with data on the property of interest. So the data from these analogs can be used to predict the property of a target substance using read-across or trend analysis. And I should note that OECD guidance document number 102 provides detailed guidance on using the Qsr Toolbox to develop chemical categories and provides instruction on data retrieval, simple analog approaches, and building chemical categories and building metabolite categories for users that may want additional guidance. So ultimately, the Qsr Toolbox can be used to predict properties. The Qsr Toolbox has a variety of Qsr models integrated, which do not require the manual identification of analogs. Examples of external Qsr models included in the toolbox include the EPI-suite models from US EPA, the Danish Qsr database predictions, and a variety of others. So ultimately, the Qsr Toolbox is a very powerful tool for bridging knowledge gaps and identifying available information. In conclusion, the Qsr Toolbox offers many possibilities to assess chemical properties with non-animal methods. It also includes a variety of Qsr models, existing data, metabolic and mechanistic information, all of which can be used to fill gaps and to scientifically support building analog and category approaches. The toolbox development is a group effort. This is managed by ECHA and OECD in a long-term collaboration and implemented by LMC, the developer, but also with substantial input from third parties which contribute to its success, not only in the form of donation of data, profilers, and models, which can be found in the toolbox repository, but also with feedback and suggestions, especially those given from nominated experts via the Qsr Toolbox Management Group. And lastly, we receive external support in the form of financing of tutorial and other development to support users implementing the functionality of the Qsr Toolbox. So I encourage you to go to the OECD website and the Qsr Toolbox site as well. There are a variety of training materials linked and video tutorials and additional information for those that may want to find more there. And I thank you for your attention.
Hello, my name is Doris Hiermann. I'm working at ECHA together with Andrea Gissi in the Computational Assessment Unit. Patience gave you an overview of the functionalities in the Qsr Toolbox, and I would like to give you a demonstration on some of these functionalities. My focus will be on how to find data and information about the properties of a substance and how to search for analogs with the Qsr Toolbox. I hope I will be able to give you also some tips and tricks when working with the data matrix in the Qsr Toolbox. It's a real toolbox with many tools, and it comes also with many options, so you have to choose from. And this begins already when you start the software, where it asks you which interface you want to use. So you have here the classical user interface. This is the main interface, and it includes all the functionalities, but it needs a bit of a training to be able to use it. And also it runs only on Windows. Then there is the simplified user interface. It's easy to use, but it includes only simple functionalities. It will be gradually replaced by the web client, and it runs only on Windows. Then there's also the web client. It is the latest interface. It is easy to use, and more functionalities are introduced at each release. The advantage is it runs on all operative systems. However, the server must be installed on a Windows machine. In this presentation, I will address in the Toolbox desktop client. I will introduce how to use the simplified user interface. I will give you a brief introduction, and there is more information in an extensive video that we will provide in the supplementary material, which takes about 10 minutes. In the classic user interface, I'm going to show you several functionalities, queries, and how to find analogs, and also finding analogs taking into account metabolism. My colleague Andrea Richards is going to give you further details on how to do a read across for each purposes later during this webinar. The Toolbox web client will be demonstrated later then by Stanislav Temelkov from LMC. In the simplified user interface, you have access to basic functionalities. You can enter chemicals here under Define. Let's use CAS number 818-61-1. Search by CAS, and it will retrieve you the chemical that corresponds to the CAS number. I confirm the selection, and then you have access to the 3 basic functionalities. So first of all, collect data. It will retrieve experimental data from the various databases from the Qsr Toolbox. Then you can apply profiling. Here you can get information about the reactivity profile of the substance. There is a huge knowledge base encoded within these profilers. This can be generic information, for example, in which database you can find the substance, or it can be also mechanistic information, for example, DNA binding, and many more. Here in red, those results are highlighted that may be of a potential concern. In order to go back to the entry screen, you have to click here, Define Goal. There is also the possibility to run specific profilers, and you can also run transformation reactions on the substance and then run the profilers on these transformation products when clicking here, specific profilers with metabolism. I don't show it here as we have prepared an example with that in the classical interface. Further, you can do basic searches for analogs by organic functional groups here, or you can choose from the various profilers here by other criteria. I'm going to show you more about these profiles in the classical user interface. Here you can identify your target endpoint. It tells the system in which endpoint you're interested in, and then it will highlight, for example, relevant databases or relevant profilers. But let's switch now to the classical user interface. This is this button here, and you can always switch back and forth between simplified user interface and classical user interface. Here you see the classical user interface. The Qsr Toolbox has 6 modules, which you see here in the top row. And in the following examples, I'm going to show you the functionalities within these modules. Remember that the Qsr Toolbox does not guide you through a workflow, but it gives you options and possibilities, like a toolbox provides tools. That means that you can use the modules in the order which you find useful, or you select the ones that you need. In the demonstration, I will address basic functionalities, but also more advanced or more hidden functionalities, as well as highlight some of the new ones. The first module is the input module. Here you can enter your chemical. You can work either with single chemicals or also with chemical lists, such as databases, inventories, or your own lists that you can define as text files. You can also here access the query tools, so you can make searches in the databases, and you can also search Euclid. And you can also define your endpoint here. So let's first start to open a new document. I would like to enter my chemical with an EC number, and for that I click here to Chem IDs. And here under additional IDs I select EC number, and I use the EC number 214-478-5. I get a morpholine derivative and I click OK. In the example, I would like to show you how to retrieve information about persistency and the mobility of the substance. As a first step, I would like to see which kind of information there is for the substance in the databases within the toolbox. So I go to the module Data. Here you can select from which databases the information should be retrieved. In this case is selected ECHA Reach. So let's gather data. Now it asks me here if I would like to get the data from all endpoints or only from specific. Because I have only one substance, I select all endpoints. If you have a number of substances, it may take a while, so you may want to restrict your search for certain endpoints. The toolbox tells me that I have here one chemical and 96 data points. I'm interested in persistency, therefore I open up here the environment, fate and transport tree hierarchy, and I see there is some data under biodegradation. I can here open up further, and I see here that there is screening data available. When you look at the cells, you can see here a small green square. You can open here to see more data behind. So the little green square means there is more data behind. Now, the organization here may not be the most convenient for you. So it starts here with percent degradation, CO2 evolution, DOC, and so on. Here is a little blue square. That means that you can change the dynamic part of the tree hierarchy with going here with the mouse over this part and press the right mouse button. And here opens up another pop-up window where you can select Set Tree Hierarchy. Here I can choose, for example, to have the test guideline up front, so I move it in the first place. You can see here now that the tree hierarchy was organized according to the guideline. And I see here I have a OECD 302B test, and it shows me 20% degradation. The 302B guideline study is an inherent test, and this low percentage indicates potential persistency of the substance. if you want to know more about the data behind, you double-click into the cell and you can access the metadata. The information is organized in an alphabetic order. So here you have information about the, for example, the year at the far right side, and also the link to the ECHA dissemination website. The nice thing with this link here is if you double click, it gets you directly to the ECHA dissemination website. You have first to confirm the terms, and here you see the endpoint study record. Now let's go back to the toolbox. Furthermore, I'm interested in the mobility, and the log KOC can be an indicator of mobility. I do, however, not see experimental data here in this field. There is a nice feature in the Qsr Toolbox that's called parameters, and it covers 2D and 3D parameters. So I would like to show you here the 2D parameters. Most calculators here are from Episode. So we are interested in KOC, so I use here the filter endpoint 3 functionality, type in KOC, confirm with enter, and I get here two possibilities. The one is based on log KOW and the other one is based on the MCI method. I have here go with the mouse on top of this cell and click with the right mouse button, and I get access to another pop-up window where I can then choose calculate my KOC. And as you can see here, I do it also for the second one. The values are low, which means there's an indication of a high mobility for this substance. Now I go back and show the whole tree again. You may wonder why there is some results highlighted here in orange, for example, for aquatic toxicity. This is a new functionality, and I find really important and great because it indicates if the test material that was used to conduct the study is not the same as the substance here. This is something specific to ECHA REACH, and I can show you a little bit more details. So here in the metadata, you find here the test material information besides the substance composition. If you double click this cell, it gives you further information about the test material. In this case, the difference is that the substance is the salt of the free acid. But especially if you want to use substances for read across, it is good to check whether the test material actually matches the substance. Under the data gap filling module, you can find Qsr models, and then you can make further predictions for the substance. Now I would like to show you something different. So we move now to the module profiling. And here I would like to show you what you could retrieve with the ECHA screening profiler. For that, we have to move here to the customized profiler. I select the P profiler and click Apply. I make the cell bigger to see all the result. And what it says here is that the substance screens potentially P or VP based on experimental data. It tells me also that BioVin predicts potentially P or VP. It says here also no Qsr predictions can be done with Vega and Danish Qsr DB. This is because the Vega module has not been updated yet to the latest Qsr Toolbox version, which is 4.5, which you can see here. And the profiler would need still to be updated. And we are thinking at the moment to have as an interim solution to remove the Vega so that the profiler work properly again. So for the experimental data, it does work, and I would like to show you how you can learn on which basis this profiler gave an alert. So you move here again with the mouse, press the right mouse button, and then it gives you another pop-up window where you can click here either view scheme about, but we are not interested. We would like to know more about the results, so we say explain. And then I select the experimental potentially P or VB result, and I click on details. And now it opens the information screen about the profiler. So what you see here is a description of the profiler under literature. And you can see here under definition the rules, how it assesses information. And on the left side, you see a decision tree. And it shows you also where it found a match. So it is this one here with the check mark. And here in the lower left window, you can see on which basis the alert was given. It's the 20.6% degradation. And this is the value that came from the ECHA REACH database. You can close the window here. So this is the example that I wanted to show you regarding persistency and mobility. Now I would like to move to the next example. For that we go again to the input and create a new document. In the next example, I would like to show you how you can find analogs to assess the endpoint mutagenicity. I enter the substance with the CAS 18463-90-6. The first thing that you may want to do is to define the endpoint. So I choose here human health hazards, genetic toxicity, in vitro, then bacterial reverse mutation assay, gene mutation, and as a test organism I select Salmonella. Okay. Now you see here highlighted in yellow the defined endpoint, and this will help me now also to choose appropriate databases and profilers. Now first I check if there is any data available for the substance within the databases of the Qsr Toolbox. I select bacterial mutagenicity database as the one suggested by the system. The system tells me no experimental data available for my substance. So next I go to profiling module, and I would like to understand if there is any specific mechanism for the target substance. Here again in green, suggested suitable profilers for my endpoint. I choose DNA alerts for AIMS and click apply. Now here it shows me no alert found. In case there is a specific alert found for your substance, you could start the search for analogs based on this profiler. So you want to find analogs which have the same mode of mechanism as your target substance. In this case, you would find a lot of substances, and it doesn't really make sense to start with the profiler that gives you no alert.
There is the option that you would, for example, search also for structural similar substances. In this case, you could also explore the rat liver S9 activation and whether transformation products have a specific mode of mechanism. So there is two ways to do that. One way is to select here under the profiling module, the simulator. I click apply. And the system tells me it found 16 metabolites. If you double click here, the structures of these predicted transformation products are depicted. Here below, it ran this DNA alerts for Ames profiler on all the metabolites, and we got a number of alerts. If you would like to know more about what is behind this, you move your mouse over this cell, you click with the right mouse button, and you can select here Explain. Now here the metabolites are sorted according to mode of mechanism. If you are interested in a specific one, you can, for example, click on the substance and tell display the chemical. Let's close this window, and I would like to show you also another way how you can retrieve the predicted transformation products after S9 activation. For this, we go here to the input module. Here on the left side, you see the substance, and you click again with the right mouse button, and you select multiplication, metabolism transformations, and then you choose the rat liver S9 metabolism simulator. What this does is that it displays all the predicted transformation products together with your parent substance in the data matrix. Now you can use again the profiling. I choose here the DNA alerts for Ames. And here now you see the parent has no alerts, but the predicted transformation products have specific types of alerts. If you want to understand what is behind a specific alert, for example here nucleophilic attack, you move your mouse here in the cell and you click with the right mouse button and you click Explain. Here I choose the alert and go to Details, and it opens up the knowledge base, the heart of the Qsr Toolbox. There is a huge amount of information coded here, and also together with the literature and background information for the reactions. I close again the window. Now if you want to search for analogs, you have to go back to your target substance, and we go to the module category definition, and instead of using here the define option to define your initial set of substances, we will use define with metabolism. For that we have first to select again the profiler, then click define with metabolism, and then we choose rat liver S9 metabolism simulator, and it gives us now options how the system should search for analogs. We are interested in the same profile for parent and metabolites, so we choose here the same profiler. And if you want to be more restrictive, so you want also the parent to have no alert, you can choose here the same profiler again. And I have now here a number of chemicals that have a similar mechanistic profile. I can show you by running again the profiler. So here for the parent, no alert found. If I also add here the simulator for predicting transformation products together with the profiler, I see here same profile for the target substance and the analogs. Now you want to have the analogs not only mechanistically similar but also structurally similar, and you see here some they look more similar, some they look a bit more different. So what you can do is you go again to the category definition module and you filter further the analogs by subcategorization, and here you can choose one of the empiric profilers related to organic functional groups. Let's choose one of the organic functional groups profilers. Here on the right side you see how your target is identified with the profiler, and on the right lower corner you see all the functional groups that were identified for the target and for the analogs, and highlighted in blue the ones that are different to the target substance. So we remove the different ones and we end up in a small set of substances which are structurally similar and also mechanistically similar in the way that they have S9 activation leading to transformation products with the same mechanistic profile. I have shown you how you can find analogs with the Qsr Toolbox that are structurally similar but also mechanistically similar. Now, if you want to use these analogs to fill a data gap via read across, I recommend that you first of all you explore here what is behind this experimental data and if the experimental data are reliable. And second, you have to make your read across case, and the Qsr Toolbox can support you with that, but certain information has to come also from the outside. For example, robust study summaries for the analogs. And my colleague Andrea Richards will later on in the webinar tell you more about the requirements under EACH. There is a nice functionality under the module report that's called Data Matrix, and it will export the data matrix into an Excel. Here you have the option to include or exclude information. And then you click here Report, and you open the data matrix, and it opens the Excel here with the information from the data matrix. Now let's go back to the toolbox. Let's open one more new document. And as a last example, I would like to show you what you can do with the query tool. So in case you have a specific structure that you would like to see in analogs, you can use the query tool or the substructure search, so one of them, to very efficiently look for substructures in the substances in the databases that you select. So let's first check which databases we have selected. So here the search would only be within the ECHA REACH database. Now let's go back to the input, and I select now here the query tool, where you can combine also searches. Here I'm interested to search for specific subfragments, but you have also many other possibilities like parameters, similarity, name, and so on. So I select here subfragment, and I add as a subfragment an ether functionality. And I add this here to the right side. And then I would like to have the substructure also to have a perfluorinated part. So I enter here a perfluorinated substructure with 2 carbon atoms. I click again OK and add. I can connect these 2 searches, and then I would like to also define a carboxylic acid functionality. Which I don't want to have in the substance. So I can say here not. And combine the 2 with an AND. So I have now logic operators between these 3 substructures. And now I can say execute or double click here. And now it will find me in the database ECHA reach all substances that have an ether functionality, 2 carbon that are perfluorinated, and no carboxylic acid functionality. Now I see here 14 chemicals were found, and they are all having an ether functionality and a perfluorinated part. So this is a very powerful way to search for structural analogs that have a specific structure. If you know that you're interested in a specific structure, or if you know that a specific structure is connected with the endpoint of concern. In these 30 minutes, I have shown you how to find information about substances in the Qsr Toolbox and how to search for analogs. There are many more functionalities in the Qsr Toolbox. If you are interested to learn more, please check out the Qsr website. Under support, you can find many tutorials. Later in this webinar, you can hear about the automated workflow for defined approaches for skin sensitization and the new web client. I hope my demonstration was useful. Thanks a lot for watching.
Hello everyone, my name is Darina Yordanova and I am part of the AOMC team that is responsible for the technical development of QSAR Toolbox. Today me and my colleague Stanislav will present you what is new in the latest Toolbox version, and we will demonstrate you some of the most important new Toolbox functionalities. But at first, let's start with some notes on the history and statistics for Toolbox. Toolbox was initiated by OECD in 2006, and the first version was released 2 years later, when actually ECHA also joined the Toolbox coordination group. The latest public version is from October last year, and an update of this version was just released. The number of active users of Toolbox is constantly growing up through the years, and here you can see the official number of registrations downloading Toolbox. However, in real, one registration corresponds to one whole company. So we estimate the number of real active users to be above 200,000 users. And here are some statistics for the downloads and download activity for the last year, showing the constant interest in Toolbox. And now, What is new in the latest Toolbox version? Here are listed some of the most important new functionalities that will be presented during this webinar. At first is the web client. Web client allows most of the basic Toolbox functionalities, including search in Euclid databases, to be done by web implementation. Euclid compatibility allows interaction between Euclid and Toolbox, and this does not only visualize the Euclid information in Toolbox, but it also allows to make different searches in Euclid databases. For example, once we have Euclid information in Toolbox, we can use the searching capabilities to search for substances having specific impurity. Furthermore, once the Euclid information is in Toolbox, we can easily see the data points which are obtained for test materials different than the main substance. Another one new functionality in Toolbox is Workflow Editor. It allows the user to implement their logic for searching for analogs or for making predictions for different endpoints. And once this information is in Toolbox, the system will apply automatically the logic. This workflow editor was also used to implement an automated workflow for obtaining skin sensitization predictions that are used for defined approaches. And these predictions are already part of an OECD guidance. But I will come back to this in a minute. Here is a list with additional new features and improvements in Toolbox 4.5, and the entire list could be seen on this link. Now, let's go back to the automated workflow for defined approaches for skin sensitization. As I said, it is already part of an OECD guidance that was published last year. In this first version of the guideline, there are 3 defined approaches that are included, and one of them, Integrated Testing Strategy version 2, use the predictions from an automated workflow in Qsr Toolbox. More about this information and different defined approaches could be found in the OECD library. And now, more about implementation of this automated workflow. As I said, this automated workflow was implemented by using of the workflow editor. And here in right, you can see the basic view of the logic that is implemented. This logic could be automatically applied to one or more than one chemical at once. However, it is not applicable to inorganics, salts, complex mixtures, etc. And what is the logic that is implemented? The logic is based on presence-absence of protein binding alerts in skin sensitization. At first, once we have a target chemical, the system starts to search, is there any protein binding alert for skin sensitization that is identified in our target chemical as parent or as a result of abiotic or biotic activation. In case such protein binding alert is identified, then the system is searching for mechanistically similar analogs, acting by the same mechanism and in the same way. After that, the analogs that are structurally dissimilar than our target chemical are removed. In case no known protein binding keyword for skin sensitization is identified in our target chemical, then the system is searching for structurally similar analogs. Once these structurally similar analogs are found, then those of them having identified the word as parent or as a result of some activation will be removed. Or finally, we'll come up with a small category of analogs that are mechanistically and structurally similar. In some of the cases, when this logic fails to make a prediction by read across, the final outcome of the automated workflow for defined approaches will be based only on theoretical knowledge. However, no matter how the prediction is obtained, either read across or as a result of theoretical knowledge only, the applicability domain will be also automatically defined. The applicability domain for automated workflow for defined approaches includes 3 different layers: parametric, structural, and mechanistic, that could be considered or not, depending on the prediction. Or, for example, if you have negative prediction as a result of read across, then only the mechanistic layer will be considered. However, if you have negative outcome as a result of theoretical knowledge only, then this means that no analogs have been found and all of the 3 domain layers need to be considered. Now, I will go directly to Toolbox and we will show you how you can apply this automated workflow and to obtain prediction for skin sensitization. Here is Toolbox 4.5, and here I have 2 chemicals loaded on the data matrix. Once you're in Toolbox, you can enter your target chemical by some of these possible ways, by class, name, structures, etc. Or you can load your own custom list. In my case, I loaded my custom list consisting of 2 chemicals. Now, I do not need to do anything more here, just to go directly to data gap filling module, where I'm seeing the button for automated workflows. Click on the button, invokes the window with all available automated workflows, including the custom ones if I have created such. Documentation for each of these workflows is available, including this automated workflow for defined approaches. It could be seen from here. And this is the view of the documentation. Let's select the automated workflow for defined approaches. Once confirm my choice, the workflow editor with the logic implemented for the selected workflow will be shown. From the section with execution buttons, I can start the logic. Because now I have 2 chemicals on the data matrix, I am seeing this message. I want to obtain prediction for both of these chemicals, so I click yes. And furthermore here, I want to see all of the steps which are done in Toolbox automatically. Because of that, I'm unselecting this box and confirm. Now the Toolbox starts automatically to apply the implemented logic. the workflow is trying to find, is there any protein binding word identified in our target chemical as parent? However, an word for protein binding has been identified as a result of activation. Because of that, the analogs will be searched, taking into account their metabolic activation. Once appropriate analogs are found, then the prediction is accepted automatically and Toolbox continue with the second chemical in the list. In this case, you can see that no word is identified in the parent structure and no word has been identified as a result of autooxidation and as a result of skin metabolism. Because of that, analogs here, such structurally similar chemicals are found, then the system will remove automatically all of them that have no word either as parent or as a result of abiotic or biotic activation, in order the analogs to be the same as our target chemical, having no word as parent or as a result of abiotic and biotic activation. Finally, the workflow shows this message that it is finished and predictions are obtained for both of these chemicals. In order to see the final results from the automated workflow, or this means the predictions along with domain information, we need to go back to this level where we have entered these two chemicals. Now, here we can see indication that the first chemical is predicted positive as a result of read across and it is in domain. However, the second chemical is predicted negative as a result of profiling. This means only based on theoretical knowledge or no appropriate analogs have been found. And furthermore, this prediction is out of domain. Let's see why this prediction is out of domain. Double-click on the result. We'll highlight the reason, or this chemical is out of domain due to the structural layer of the domain. Let's see more details. I'm explaining this. And now I can see that my target chemical has above 91% fragments that are known for the system. However, there is a small percentage of fragments that are unknown for the automated workflow. Or this means that there are no chemicals in the training set of the automated workflow having such fragments. Let's see which are these fragments. If I go to unknown fragments, I can see exactly which are the fragments that do not present in any of the training set chemicals. With this, I conclude my presentation. Thank you for your attention.
Hi, my name is Stanislav. I'm a developer at Laboratory of Mathematical Chemistry, and I'll take over from Darina, showcasing the next thing that is new in Toolbox 4.5, the web client. As a version 4.5, the web client comes bundled with the Toolbox installation. And in fact, in the latest version of 4.5, Service Pack 1, you will find the updated Toolbox web client version 2.0. I'll talk about potential users, new features, and deployment scenarios. Now, if you've never used the web client, or if you are wondering if it fits into your workflow, let me tell you how we see the potential user groups. The first potential groups are the new users, those who are unfamiliar with the Toolbox. These users can use the web client to get familiar with the basics without being overwhelmed by the full interface. The next group is users of the Simplified Ui. The Simplified Ui is an alternative shell which can be used for everyday tasks without the complexity of the full version. If you've already been using the Simplified Ui, then you can safely switch to the web client to accomplish the same tasks. And then there are the regular users who are already experienced with the desktop version, but they prefer to use a lightweight client in a browser, regardless of the operating system, or to utilize the benefits of a shared deployment, which I'll talk about in a minute. Version 2.0 of the web client brings two major new features. The first one is a real data matrix, which is the key interface element of the toolbox. On the matrix, chemicals are ordered into columns, results are ordered in groups, and they are all in an endpoint tree. This data matrix allows reordering of columns, resizing of columns, collapsing rows, and so on. The second new feature actually couples two sub-features. This is the ability to fill in data gaps by running QSRs or automated workflows, the same as the ones Darina talked about. I'll show this during my demo. Apart from the new features, version 2 also packs some important improvements, such as the ability to import structures with a 2D editor, selecting multiple chemicals during the inputs, improving the display of data points, richer explain service, and a better export as well. Let's talk about the two main scenarios in which you may use the web client. The first one is an alternative way to access your own Toolbox server. To do so, we've added the so-called web suites, which is installed during the Toolbox installation. Once launched, from it you can access a web client, which communicates directly with the Toolbox server and the database that is installed on your own computer. The second alternative is using a shared server. In a company, agency, intranet, you may have the Toolbox installed on a central server, and then users can access it using either a desktop or a mobile device. Now, I'll switch to demonstrate you what the current web client looks like. Okay, so once you install Toolbox 4.5, the installation brings you the Toolbox desktop client, the server, and now you also get the web suite. Launching the web suite brings up this window from which you can access the Toolbox web client and the web API. Clicking on the web client link loads the web browser connected to your own Toolbox server. Now, here on the left, all the commands are grouped in these menus. And if you're wondering where to start, we've made it obvious by placing this big button here. Now let's add a chemical to start working. You can add a chemical by CAS number, by EC number, by chemical name, or by structure. Let's enter a chemical by chemical name. I'll enter dichlorophenol. We get 3 hits. Let's select them all, confirm, and then we have in the matrix those 3 chemicals. If you expand the structure info, you can browse basic identification information for all of these chemicals. Now, if you look at the other section, they are all completely empty because we haven't done any work with these chemicals yet. What we can do is to calculate some parameters. Wherever you see this flash icon, this means that clicking on this command will trigger it immediately and there will be no questions asked. Let's click on it. This automatically calculates all the parameters for those 3 structures, and you can now browse them, unfolding the 2D and 3D sections. You see we have the values for each of the chemical. Clicking on some of the parameter names brings more information about the calculator that has performed the calculation or the Toolbox object. So for example, this one here, this is calculated by a module that has been authored by the US EPA. Now, I'll remove some of the chemicals to make room for more information. And let's move on to the next sections. Under profiling, you can again either run all of the profilers or you can select specific profilers. Clicking on this button brings up a window in which all profilers in the Toolbox installation are listed. You can filter them by type, or you can type a name to filter the needed profiler. Let's select all endpoint-specific profilers, select them all, confirm, and now the system has calculated those profilers and we can browse the results. Clicking on a result brings literature for this profiler, which is the same that users may be used to in the web client. Clicking on a profiler name, again, as in the case with the calculators, which brings some information about the Toolbox object that has performed this task. Now that we have seen how we can retrieve parameters and profiles, let's move on and collect some data. Collecting data can be accessed in the same two ways. We can now either retrieve all the data available in the Toolbox server, or we can collect specific data. To collect specific data, we can select the endpoint from the endpoint tree, but let's go and collect all data available for this chemical. During the collection of data, you'll see this progress bar on the top, and when it's done, you'll see that next to each of the sections of the endpoint tree, you have a label which displays information for how many chemical and how many data points we have on this row. For example, let's open physical-chemical properties. We see we have data for boiling point, for melting freezing point, and so on. Clicking on the data brings some meta information about the data point. Again, you can also simply type in the filter and you'll see the point of interest. Next, let's go and look for some analogs of this chemical. You can do this by either a functional group, which uses the organic functional groups and selected databases, or you can specify the exact profiler and database that you want to use. Let's do that. First of all, we'll look for skin inclusion rules, skin irritation corrosion inclusion rules by BFR. We can select this profiler, confirm, and then you need to select the database in which we are going to look for chemicals. In my example, this will be the skin sensitization ECHA. Let's confirm. And the system informs us that it has found 4 analogs, and it will load those 4 analogs. From this point on, you can continue working with those chemicals, retrieve data for them, run workflows or QSRs on all of them. To continue with my demonstration, I will start with an empty matrix, and I want to show you how you can add a chemical using the 2D editor. So again, you go to the input section, and this time use the structure. From here, you can either type the smiles code directly, or you can launch the editor and then use the tools here to draw a chemical, or again, by simply pasting a smiles, the system will draw it for you. Click OK, then search as in the previous example, and this brings one chemical as an example. We'll also add one more, which will be the same dichlorophenol, but this time I'm going to select one of those chemicals, this one. Now I want to show you one of the 2 ways that you can fill in data gaps. The first one is by running workflows. Running workflows is accessed by this command here, which shows all the available workflows on the Toolbox server. These are the workflows that come pre-installed with the Toolbox, and if you have already created new ones, then they will show up in this dialog as well. Let's launch the workflow for defined approaches. Right now, the execution is done on the server, and if you're using a remote server, which is powerful enough, then you may safely do this from a mobile device or from a less powered notebook. As you see, the system is now simply waiting for the result, which is now displayed here under human health hazards. We have sensitization, the result for the first chemical is negative, out of domain, and we can see why it is out of domain for this workflow. And for the next chemical, the result is positive. The last thing that I want to show you is how you can run QSRs. Let's go to the next section, open the Qsr Editor, and here you'll see that QSRs are grouped under the endpoint tree. So let's go on the human health hazards, again sensitization. We see a number of QSRs available here. Double-clicking on any of those, we'll put it in the list and we can apply it. Clicking on apply will bring up this window because as part of the QSR database, you need to read this license and confirm it. Once you run it, you get the result, which is positive in domain. The last thing that you can do is export either the chemical or the entire matrix. Searching the Euclid database will be presented by Andrea in the following presentation. Thank you for your attention.
Thank you for joining us today. My name is Dr. Martin Phillips, and I'm a chemist at the United States Environmental Protection Agency's Office of Pollution Prevention and Toxics. I'm joined today by my colleague, Dr. Brianne Ricoeur, who recently joined the agency from being a professor of pharmaceutical sciences. Today, we are going to talk about the OECD QSAR Toolbox, its use at the Environmental Protection Agency, and its connection with Oncologic, which is an expert system for predicting carcinogenicity. Oncologic is based off of the biochemistry that underlies carcinogenesis. So carcinogenesis is recognized to be a multi-step, multifactorial process that requires both genetic and/or epigenetic cellular changes to progress to invasive cancer. Oncologic uses the three-stage model of carcinogenesis: initiation, promotion, and progression. In initiation, you have gene mutations or epigenetic changes that lead to alterations in DNA. Promotion leads to a clonal expansion of a cancer phenotype, and progression is where you get metastasis. Chemicals can interact at any stage of this three-stage model. Some carcinogens will act on one stage, such as initiation. Very potent carcinogens can act on all three stages. Oncologic is a mechanism-based expert system for predicting carcinogenic potential. It was developed by domain experts at the EPA, including David Lai, Yin-Tak Wu, Mary Argus, and Joseph Arcos, in collaboration with an expert system developer. And it incorporates knowledge from structure-activity relationships on over 10,000 chemicals. It uses a class-specific approach to optimize predictive capability. What that means is that the rules for predicting cancer probability are different for an aldehyde versus an aromatic amine versus a saltone. It considers all relevant factors, including biological input when possible, and that's going to be part of what my colleague Dr. Ricoeur covers later. So it's not just structural information, it's also bioassay information, can go into the predictions. The resulting report provides both a detailed scientific rationale and an ordinal ranking from low to high concern. And in January 2021, Oncologic version 8 was migrated to a newer standalone platform, Oncologic 9. The initial migration covered the organic chemistry module, which again is what we're going to focus on today, but there are plans to move the other modules, which cover fibers, metals, and polymers, to the new platform. So how does Oncologic work? It mimics the thinking and reasoning of human experts using knowledge-based rules for chemical classes to predict chemical concern, cancer concern. So it assigns a baseline concern ranging from low to high for each structural class. So, like I said, aldehydes and aromatic amines will each get their own baseline concern level, And then it starts looking at the substituents around the molecule. Each substituent can change the concern level. You can either increase the concern level or decrease the concern level. We call these rule makers and rule busters. And some of them have a neutral effect on the concern level. So primary chloride, alkyl chloride might increase the concern level since you're adding an additional reactive group. A methyl at a particular position may have no effect, and then there could be other substituents that would actually lower your concern level, like a long alkyl group that maybe makes it less bioavailable. So there are multiple benefits from using this software. It helps expedite the decision-making process by speeding up the rate at which decisions can be made through the use of computational tools, allows sharing of knowledge between assessment teams and amongst agencies or companies. It helps reduce error and inconsistency just by being a coded system, and helps formalize knowledge rules for cancer hazard identification. Oncologic 9 isn't static. We can make adjustments as new science becomes available. And now that it's been ported over into a more modern system, the agency is certainly interested in any suggestions people might have to incorporate, even more knowledge about carcinogenicity into the software. And finally, it allows non-experts to reach scientifically supportable conclusions. The final report that's generated is quite detailed as far as providing the scientific justification and rationale for the concern level. So what are those concern levels? Like I said, it's an ordinal ranking from low to high. So low is unlikely to be carcinogenic. Low moderate is likely to be weakly carcinogenic. Moderate is likely to be a moderately active carcinogen. Moderate to high is highly likely to be a moderately active carcinogen, and high is highly likely to be a potent carcinogen. So the low moderate acts usually at one stage of the three-stage model, like initiation, and high acts at all three. The last concern level is marginal, which is likely to have equivocal carcinogenic activity. So there are quite a few cancer bioassays in rodents that have given equivocal results. Maybe it's only positive in a single sex or a single species, or tumors were noted, but there was no dose response. So that's how that information was incorporated into Oncologic. So if you were to test something with a marginal concern level, you would likely get equivocal results in a 2-year cancer bioassay. Okay, so let's talk a little bit about how it's being used in the new chemicals workflow. So Oncologic helps to refine class-based structural alerts to better predict cancer hazards. We start with a company that's making a submission to the US EPA starts with their new chemical substance. If it's never been manufactured or imported into the United States before, they make a submission to the agency called a pre-manufacturing notice. That kicks off a series of discipline-specific reviews. There are 6 disciplines: chemistry, fate, exposure, engineering, ecotoxicology, and human health. So the chemists go first and validate that it is a new chemical substance, and then the other 5 reviews are run more or less concurrently. In the human health team, we might flag the new chemical substance for a structural alert, like if it's an aromatic amine. We know some aromatic amines cause cancer, but not all do. So the purpose of Oncologic is really to determine not whether aromatic amines generally have cancer concerns, but whether the new chemical substance specifically has cancer concerns. So we can enter the structure via SMILES, which is just a text string that represents the structure, and is generated by our chemists here at the EPA. Into Oncologic version 9. If you end up with low concern, then the analysis is done. So again, the concern levels go from low to high. So if it's low, then we can basically finish up with the cancer part of the assessment. If we get a different result, then what our team will do is perform an analog search to look for slope factors so that we can perform a quantitative risk assessment or weight of evidence analysis. So a low result is actually relatively common, and it can help save our team time in eliminating the need for an analog search. At this time, I'm going to pass it over to my colleague, Dr. Ricord, to give you the rest of the presentation.
Thank you, Dr. Phillips. As mentioned by Dr. Phillips, Oncologic 9 uses structure-activity relationships and biological input to assign a concern level for a specific compound. For some chemical classes, the biological input comes from a series of interactive questions that are asked by Oncologic that require the user to have access to experimental data and/or expert knowledge, which may hinder an accurate prediction of concern. An example of this class of compounds that needs biological input for Oncologic to predict the concern level are the organophosphates. And the organophosphates are of concern because they are a diverse structural class that we know can have a wide range of carcinogenic potentials, depending on the specific compound of interest. Therefore, the aim of this current project is to reduce the need for expert knowledge through the integration of QSAR Toolbox with Oncologic 9. This should enhance the usability of Oncologic 9 through the reduction of the need for expert knowledge and increase the consistency of the predictions of carcinogenic potential. We can take advantage of the experimental data that exists in the databases contained within QSAR Toolbox to provide answers to the interactive questions. QSAR Toolbox can automatically answer questions related to predicting concern for organophosphorus compounds. And the questions that we can automatically predict, I will show on the next slide. We have integrated QSAR Toolbox with Oncologic 9 from work with our collaborators at OECD and LMC. The 4 questions that can be integrated and answered automatically by QSAR Toolbox are: select the results of the in vivo genotoxicity testing, answers positive, negative, or unknown. Select the results of the in vitro genotoxicity testing, answers positive, negative, or unknown. Is the chemical highly toxic? Yes, no, unknown. Is rapid hydrolysis or detoxification expected? Yes, no, or unknown. Prior to the integration, all the questions on the slide required the user to have knowledge of experimental data for the specific compound of interest and/or expert knowledge. The users may have defaulted to answering unknown to all questions, and therefore the prediction of concern may have been hindered. This integration, now through the automatic answering, allows the possibility that answers can be inputted into the system for a better prediction of concern. So how does QSAR Toolbox answer these questions automatically and input the data into Oncologic? We're going to start with the classification for genotoxicity. QSAR Toolbox databases provide in vivo and in vitro genotoxicity data through gene mutation studies that are contained in its database for numerous compounds, or chromosomal aberration data that are contained in its databases for numerous compounds. The system provides an automatic answer based upon the extraction of results that it searches for in its databases. So the answers will be based on if the compound has been tested and positive genotoxicity experimental data is found. So that means at least one positive experimental data point was identified. Then Oncologic will answer that question as yes, positive data. QSAR Toolbox will say, we found a positive experiment. Answer, yes, there is positive data. If only negative data was found, the compound was tested, but only negative data was found for the gene mutation or chromosomal aberration studies, a negative response will be given to Oncologic from QSAR Toolbox. If the chemical was not tested, and QSAR Toolbox cannot find any data, so it will be seen as unknown, or perhaps even the data extracted may have been technically compromised, inconclusive, Qsr Toolbox will report unknown back to Oncologic. The answer is explained in the final report in Oncologic and justified in Qsr Toolbox. Here are the databases that are available from Qsr Toolbox, and I have provided links to the relevant databases for search purposes. And this slide just shows the distribution of data across the databases that Qsr Toolbox uses to extract the data. You can see that ECHA Reach has data for over 10,000 chemicals, and Genotoxicity Oasis also has data for almost over 8,000. For the classification of toxicity, Oncologic 9 incorporates toxicity data in its overall classification for level of concern. A more highly toxic compound will be downgraded because the compound doesn't need to kill the cells to be considered carcinogenic. It needs to actually just cause these gene mutations or work on those levels that Dr. Phillips talked about. In order to answer this question about being highly toxic, Qsr Toolbox will extract acute toxicity data. And what we are talking about in acute toxicity data, referring to experiments that looked at adverse effects occurring following oral or dermal administration of a single dose of a substance or multiple doses given within 24 hours or inhalation exposure of 4 hours. The specific data that Qsr Toolbox will be extracting will be looking for the LD50 or LC50 values from these acute toxicity experiments. The LC50 or the LD50 is the amount of chemical need to cause death in 50% of the animal population. Again, here are the Toolbox databases that Qsr Toolbox will search, and here is the distribution of data that we find across these different databases. The acute oral toxicity database and ECHA Reach both having data for over 10,000 chemicals that are able to be searched. Now, how does Oncologic provide an answer of yes, no, or unknown to, how does Qsr Toolbox provide an answer of yes, no, or unknown to Oncologic? We have decided to use the designation of highly toxic based upon the system that's used by the globally harmonized system of classification and labeling, the GHS. The GHS has 5 categories for classifying acute toxicity. 1 is most toxic, 5 is least toxic. We set our highly toxic as category 1 based on ranges of LD50 or LC50 values. For example, if Qsr Toolbox extracts data for a compound that was orally administered and with an LD50 less than or equal to 5 milligrams per kilogram, an answer of yes will be reported. Any value higher than that 5 milligrams per kilogram will be reported as a no. If the compound wasn't tested and no data can be found, an answer of unknown would be answered. As you can see, the cutoffs change based upon the route of exposure. So the cutoffs for dermal for high toxicity versus that for the different routes of inhalation are all different. And again, in all cases, if the compound was not tested, an answer of unknown will be given. The last question that we're going to talk about the automatic answer for is the question about rapid hydrolysis or detoxification. Rapid hydrolysis will decrease the overall level of concern because, again, the compound would be expected not to stay in the body and would decrease its ability to cause the carcinogenesis to occur. In this case, Qsr Toolbox can search for experimental hydrolysis half-life data, but if data is not found, it can also model a hydrolysis half-life for the specific compound. So not only can we use the data that's in Qsr Toolbox to look for the half-life, but we can also avoid an answer of unknown by using modeling. Of course, if the modeling fails and no data is found, an answer of unknown would still have to be reported back to Oncologic. However, if we find data or the modeling works and we can classify the chemical into the following categories based upon its hydrolysis half-life. As you can see, yes would only be extremely fast, or what we're saying very fast. So this would be hydrolysis half-life of less than 1 day or 1 to 5 days. And this would be reported back to QSAR Toolbox. Anything greater than this would be a no. These values were chosen quite arbitrarily. And as more data comes in about the use of these particular values, we will reassess and perhaps change what our cutoffs should be. I'm now going to show you how Oncologic and Qsr Toolbox work together to show a complete, transparent picture of how a prediction for level of concern was given. So this slide is showing what you will receive from Oncologic 9 after you've entered a compound. I've entered an organophosphorus phosphate compound into Oncologic and asked it to predict its level of concern. In this case, I used an alkyl phosphate because these compounds are commonly used in flame retardants and are of interest for their potential carcinogenic potential. So we received a level of concern for this particular alkyl phosphate of moderate, and its justification is based again on structure-activity relationships, which Oncologic is showing you here, and biological input. 2 questions had to be answered, automatically answered by Qsr Toolbox to get the final level of concern. And we're going to focus on how we can see how Qsr Toolbox automatically answered these questions. In order to do this, we will focus on the right portion of this slide. We will first hit the Explain Answer button, which pulls up this window, which says select the question to explain. We're going to select the results of the in vivo genotoxicity testing, and we're going to hit OK. This will automatically pull up Qsr Toolbox. Qsr Toolbox will automatically open up with the explanation for why a positive answer was given for Oncologic for the what is in vivo genotoxicity reported question. And this is the full explanation. We're going to go through this pane by pane. And as you can see right here, positive in vivo data was found. And how do we know this? Well, on the next slide, I can show you that Qsr Toolbox is told to look for data that's requiring the chemical to be positive in one of 2 genotoxicity scales that's embedded in the toolbox. It is searching gene mutation data and chromosomal aberration data. So it's combined by this or gene mutation or operator chromosomal aberration. If positive data is found in either of these 2 studies, a positive answer will be given back. And in this case, no positive data was found for gene mutation studies. See the big red X. However, there was a positive chromosomal in vivo aberration study found, and we see that green check. Now going back to that whole panel that shows the complete explanation, if we focus on the bottom, we can see how Qsr Toolbox arrived at this result. These are all of the databases that Qsr Toolbox searched to look for genotoxicity data. And you can see all of them listed. All of the available data is listed here on the right in terms of numbers. You can see 280, 11. This is all of the data that it found searching all of its databases. And if you look down here, we could see the different types of methods. If you expanded this, it would say we're searching for in vivo. If on the left, we can find the exact study that yielded this positive result. So the user can get the study, look it up on their own and verify for themselves that this is in fact a positive data point for in vivo genotoxicity. And we can see that all of this is fully explained in this explanation. So Using Oncologic and Qsr Toolbox together, you get a complete picture of why a concern level was predicted, and also verify for yourself the data that was used to give the positive answer. Now, I'd just like to talk about some very exciting recent updates that I'm not presenting on today, but we will look forward to talking about in the future. There is a new version of Oncologic 9.2, and it has even more capability to answer these interactive questions. We can now automatically predict the alkylating ability, so the DNA alkylating ability for an organophosphorus compound, and automatically predict the chelating ability for a specific organophosphorus compound using data that is contained within the Qsr Toolbox. There is also now an Oncologic 9 add-in for Qsr Toolbox. This allows the evaluation of the carcinogenic potential of multiple compounds using batch mode within the toolbox. The Qsr Toolbox and Oncologic 9 automatically can work together to extract information from Qsr Toolbox to automatically answer Oncologic 9 questions. And both programs will open and work together simultaneously. And last, there is a Qsr Toolbox update, Qsr Toolbox 4.5 Service Pack 1. I'd like to thank the USC, ECHA, and LMC for their work on Oncologic 9 and the integration to answer the automatic questions that I presented today. And of course, you for your attention. If you have any questions, please feel free to email me or Dr. Phillips at these email addresses. And thank you.
We will now see the link between the Qsr Toolbox and Euclid. Euclid is a database for chemicals and their properties implementing OECD harmonized templates. Data for rich substances are submitted in this format. Euclid is now connected with the Qsr Toolbox, and this is done via a plugin. What the plugin does is that it allows the import of structure and endpoint data from Euclid to the Qsr Toolbox. When the data is in the Qsr Toolbox, they can be, for example, searched, and then the search results are provided by Toolbox also as links to the Euclid documents in case you want to go back to your Euclid source. But then you can also run all other functionalities of Toolbox and its workflows to generate knowledge and predictions. And also this information can then be imported in Euclid as endpoint study records. If you are submitting a rich dossier, the endpoint study records will be manually compiled and not just include the information straight out of the Toolbox. The Qsr Toolbox plugin for Euclid has been developed to provide a tool to perform structural searches at composition and test material level in Euclid data, as requested by the OECD Euclid user group. Even before the plugin, the Qsr Toolbox was able to connect to Euclid, but with only limited functionalities. First of all, you need to download the plugin and install it. You can do it from the address written in the slide in the Qsr Toolbox website. The plugin is called TEDRA, which stands for Toolbox Extended Data Retrieval and Aggregation Tool. And I will save you from all the technicalities, but what you need to know is that the plugin is a file that needs to be downloaded and copied in the Euclid server installation folder. In this link, you will also find detailed instructions on how to do it. After you have installed the plugin, you can now connect the Toolbox to your Euclid database. And you do that from the data module in Toolbox there is this Euclid 6 functionality, and this will prompt the connect to a Euclid server window where you have to use your Euclid credentials to connect. After you are connected to your Euclid database, you have to choose if you want to import only structural information or also endpoint study records. In case you decide to also import Euclid endpoint study records, you can filter them to limit the import to only the information that is relevant for the toolbox. For example, only experimental data or only reliable data. The first time you import a Euclid database, especially if it is a large one, it can take a long time. The whole ECHA rich database takes, for example, 1 or 2 days to be imported. But after the first import, it's possible to do incremental updates that make the time needed for the update much, much shorter. From now on, I'm going to give you some examples of the functionalities related to Euclid in Toolbox using ECHA-Rich as an example. And remember that ECHA-Rich data are Euclid data that are pre-imported in the Toolbox. So to work on them, you don't need to install the plugin or to import manually the data. But all the functionalities that you are going to see in this demonstration are also available for databases in Euclid format that you may import yourself. The only thing that will not be available are the links to ECHA public chemicals database. Before we see the example, I want to spend a few words about ECHA-Rich database and what it is, what it contains, and so on. First, it includes published data from rich registrations. Qsr Toolbox only imports structural and endpoint data. The structural information comes from Euclid section 1.1, so-called identification, section 1.2, the composition, and from test materials reported in each endpoint study record of rich dossiers. The compositions that you import include constituents, impurities, and additives. Remember that there could be multiple compositions for each registered substance. Impurities are published and therefore included in the ECHA-Rich disseminated database only if they are relevant for classification. And then another aspect that is among the databases in Toolbox is exclusive of ECHA-Rich is that for some data in ECHA-Rich, the test materials in endpoint study records can be different from the registered substance, for example, in cases of read across. And this is what Doris Hierman has demonstrated in her previous presentation, that data that are from test materials different from registered substance should not be used for read across purposes. Now that we know about ECHA-Rich, let's see what information is included in Toolbox. First of all, you can access detailed Euclid substance identity information. You can expand the structural info tab and then click on the sources. And if one of these sources is a Euclid database, then you will have the link to ECHA public chemicals database in the case of ECHA-Rich, or the link to your own Euclid database. And this you can access by clicking on the button with the Euclid logo. And then if you click on the button with the I in the blue circle, then you can access composition details and even multiple compositions when they are available. Now let's talk about the Euclid searches. As said, the starting point is to import Euclid data in a Toolbox using the Toolbox plugin for Euclid. And remember, this step is not needed for ECHA-Rich data because they come pre-imported in Toolbox when you install the software. Then you can search the structures with the Toolbox using either the web client or the classical client, and this will be the subject of my demonstration in a moment. And then you can visualize the search results in Euclid, because the search results in Toolbox include link to Euclid documents. And exclusively for ECHA, the Euclid links are replaced by link to public information or each registration that is on our website. So we will see how to perform the search in the desktop client from the input module. And remember, the search in the desktop client allows you to use a little bit more advanced functionalities. For example, to define concentration ranges at which the substances you are searching for are present in the composition of the substance, or also to define more than one criteria per search. Or you can search the Euclid data using the web client. This client is more user-friendly. I think it's easier to use, but does not allow very complex searches that include composition, concentration details, and so on, but still allow you to define the scope of the search by limiting the databases you want to look at, the entity, if it's a dossier and or a substance, or substance type, and the Euclid sections you want to search. And with this, we can now move to the practical demonstration of the searches. In this demonstration, we will see how to search Euclid data in the Qsr Toolbox web client. When you open the web client, you are presented with this window, and the Search Euclid Database function is available with the red button on the bottom left. After you select it, you have the possibility to input your search and to define the scope of the search. First, you can specify if you want to input the structure as a fragment or as an exact match, or if you want to use a cast number, EC number, or a chemical name. Second, you can define the scope of your search in terms of databases. I have not imported any custom Euclid database, so the only one available is ECHA, which comes with the Toolbox installation. Then you can define the type of substances you want to search. If you want to look at both datasets and or dossiers, ECHA all includes dossiers, so it will not matter if dataset is included or not. And finally, you can specify which section of the dossier to search. For those of you that are not familiar with Euclid, it could be a bit confusing to understand the difference between searching the identification section for the representative substance or the composition. But after this demo, it should become clearer. So, to start, we can define the match, exact match of the formaldehyde structure. You can input by typing the smiles, or you can also use the draw functionality. If I search for formaldehyde only in the representative substance, I should get one hit. And this result is the dossier of formaldehyde, where indeed formaldehyde is the representative substance. If I search for the same structure, but as a constituent, then I should find not only the dossier of formaldehyde, where formaldehyde is obviously one of the constituents, but also all other substances that in their dossier include formaldehyde as a constituent. In the search results, I have the links to the Euclid dossier and the substance in Euclid that matches the search criteria. However, these links do not work for the ECHA database because you don't have the Euclid database behind ECHA, but it will work if you have a custom Euclid database. While the link in the middle, which is only enabled for ECHA data, links to ECHA dissemination web page, and we will see that in the composition of this substance, formaldehyde appears as one of the constituents. Similarly, If I search for formaldehyde, not as a constituent but as an impurity, I will see different search results, where formaldehyde should indeed appear as an impurity in at least one of the compositions of the dossier. Finally, I would like to show you that you can also search not for the exact structure, but just for a fragment. As an example, I can search for isothiocyanate, and again, I'm just going to type the smiles because it's faster, but alternatively, I could draw it. I'm going to search for it as a representative substance, and then I will see all substances in a rich database that include this fragment. And when I click to visualize the structure, I can indeed see that this is a fragment of a bigger structure. Finally, I could search for structures in the test material section. Now, the ECHA-rich database has millions of test materials, so this search can be very slow. Furthermore, in the current version of the web client, when the hits are found, it is not specified in which endpoint study record the test material is found. So this functionality is a bit limited in its use now, but we are going to improve it in the next release. Now we will see how to perform a Euclid search in the QSAR Toolbox desktop client. First, go to the Input module, under Search, select the Euclid search. Select the database you want to search in. I don't have any custom Euclid database, so I can only select ECHA-rich, which comes pre-installed with the toolbox. Then, if you want to search the registered substance, you have to put the search criteria here. And you can again select either a fragment or an exact match, which is called the smiles here. I'm going to type the smiles of formaldehyde, but you can also draw it and perform the search. I see one result, which is the dossier of formaldehyde with the information. Alternatively, if you want to search for the same structure but present in a constituent rather than registered substance, I need to add a component, select constituent here, draw the structure I want to search, specify that it is an exact match, and search. And now, in addition to formaldehyde, I also see the other dossiers where formaldehyde is one of the constituents. In the structure info, in the identity, I can also see the corresponding link from the dissemination website, where I can see that formaldehyde is really one of the constituents of the substance identified by the tool. Or I can view the details to check within the toolbox itself the composition information of this substance and see that formaldehyde is one of the constituents. In the desktop client search, I can also specify more details, such as the concentration range at which constituents, impurity, or additives have to be present, or specify more than one component at a time that must be present within the composition of the substance. Note that for these concentrations to work, the database that you import must have information on the concentration of the different constituents, which is not the case for ECHA-rich. Finally, I want to show you that in the Toolbox desktop client, after you have retrieved the search results, you can also go to Data and collect the data for the results you have found. If you are interested specifically to the rich data, you select only the ECHA-rich database, click on Gather. You can select all endpoints or select which endpoints to read. And then you will see all different data for the substances that matched the query.
Hello, my name is Andrea Richards. I'm a scientific officer in ECHA's Hazard Assessment Directorate. I will show you in my presentation how the Qsr Toolbox can support read-across submissions under REACH. First, I will give you a short overview on how the toolbox is used for REACH submissions. I will also explain the requirements for successful read-across under REACH, and then give some more examples on how the toolbox can be used to support read-across limitations and best practices. And at the end, I will summarize the take-home messages. The toolbox is widely used in REACH dossiers for all types of users. It is mentioned in over 2,000 REACH registrations. What is the toolbox used for by registrants? First of all, regarding read across, to identify analogs and find experimental data to read across, to find or generate information to support the read across hypothesis, for example, to support the claimed similarity between sources and target. And we are seeing often standalone predictions using trend analysis and read across results just submitted with the automated report from the toolbox without additional justification. And this is generally not sufficient. Other uses concern predictions from external Qsr models integrated in the toolbox. And often we also see toolbox profilers submitted as predictions for an endpoint. And then there's the new use within the integrated testing strategy of the defined approach for skin sensitization. I will briefly make some comments on the latter uses before concentrating on read across. Qsr Toolbox includes external Qsr models, such as, for example, Apple Suite models. And in the dossier evaluation, these results are then assessed against the criteria for QSARs. These criteria are laid down in REACH Annex 11, Section 1.3, and the corresponding ECHA guidance. More information is also available from a previous ECHA webinar as linked on this slide. However, profiler results, basically structural alerts, cannot be used as Qsr predictions to fulfill the REACH standard information requirements. For example, the lack of mutagenicity alerts does not allow to conclude that a substance is not mutagenic. Profilers generally do not fulfill criteria for valid Qsr models, such as defined applicability domain or appropriate measures of performance. They should rather be used for identification of analogs. We have already heard in a previous presentation of the webinar about the automated toolbox workflow for skin sensitization hazard prediction. It can be used in the version 2 of the integrated testing strategy for defined approaches for skin sensitization. This has been described in the recent OECD guideline number 497. Following this guideline, and if all results are within the applicability domain, etc., the defined approach can give a valid conclusion on skin sensitization. So the toolbox has an important contribution for this new guideline. Looking now at how the toolbox can support read-across approaches. First of all, the toolbox is one of the best tools for identification of analogs. It's based on structural and mechanistic similarity and also includes considerations on metabolism. The toolbox is also very helpful for finding source substances which have available experiment data that then can be read across. And the data in the toolbox database include metadata linking to the original sources when available, so it's possible to look up more details of the studies. The toolbox can provide different other types of information that can be used to support the read-across hypothesis. For example, to support the similarity of sources and target with structure and mechanistic information. I will give some more examples later. However, it's important to note that a proof of similarity cannot be based only on profilers from the toolbox. The toolbox can fill data gaps by predicting properties from analogs by trend analysis. For example, when there are many data points available for an endpoint which correlates with a physical-chemical property, such as log KOW, and follows a trend. Or it can fill data gaps by reading across study data from the source substances. We see very often that results from the toolbox submitted in REACH dossiers are flagged as QSARs. Maybe it's due to the name of the QSR Toolbox. However, this is not correct for cases where the results rely on data from analogs. Then, in reality, it's read-across. And the results are being assessed according to the requirements for read-across at ECHA. This might not be the case for trend analysis for environmental endpoints with many analogs, which can be considered as QSARs. And generally, it should also be said that there are some limitations of the toolbox. The toolbox does not always contain all experimental details, for example, kinetic data such as rate and quantitative considerations of metabolism. Therefore, additional information needs to be searched from other sources as well. In the following, I will explain the requirements for read-across under REACH to understand what kind of information and what details are needed for the read-across also by using the toolbox. The information requirements under REACH depend on the per annum tonnage band of manufacturer import. The standard information requirements are laid down in the REACH Annexes 7 to 10 and refer to relevant test guidelines such as guidelines from the OECD. And these standard information requirements can be adapted, for example, according to specific rules applying for the different endpoints, or according to the general rules listed under Annex 11, which includes read-across adaptations. Section 1.5 of Annex 11 defines these read-across adaptations. A group or category is defined as containing substances which are likely to have similar properties as a result of structural similarity. By read-across, effects of a substance in the group can be predicted using existing data from other substances within the group. The similarity might be based on common functional groups, common precursors and/or breakdown products, or a constant pattern in changing of potency. There has been a recent update of REACH annexes to clarify the information that needs to be submitted, also for read-across in Annex 11. Overall, the information needs to be adequate for the purpose of classification and labeling and/or risk assessment. The results should cover the key parameters and the exposure duration of the study that should normally be performed for an information requirement. And it was specified that the adequate and reliable documentation should include robust study summaries for each source study, an explanation why the properties of the target substance may be predicted from other substances, and supporting information to scientifically justify the hypothesis and prediction. There are a few specificities of REACH, which I want to mention. Under REACH, the substance is not limited to one chemical structure, but the substance can contain constituents, impurities, and additives. Even if structural similarity is a basis for read-across, structural similarity alone is not sufficient to justify a prediction, but a specific hypothesis is needed which links the structural similarity to the effects predicted. This also means that the read-across prediction is endpoint specific. The hypothesis might be different for different effects, which can have different biological targets, etc. And last but not least, not the conclusion of a study should be read across, but the complete study with all results. One very important document related to read-across under REACH is the Read-across Assessment Framework, the RAF. It has originally been compiled for ECHA evaluators to allow a consistent and transparent assessment of read-across submissions, but then it was made public and it can be used by registrants to improve their read-across approaches. The RAF structures expert judgment of complex scientific questions on critical aspects of read-across. For this purpose, the so-called assessment elements have been defined. They describe scientific considerations deemed crucial to judge validity and reliability of read-across. There are common assessment elements for analog or category approaches, and there are scenario-specific assessment elements, both for human health and environmental or fate effects. Overall, going through the assessment elements provides guidance for structured analysis of reader cross submissions and justifications. The RAF can therefore identify strengths and weaknesses of a reader cross approach and of the justification, and it can help to characterize the confidence an assessor can have in the proposed reader cross prediction. These assessment elements include, for example, the identity and characterization of source substances, the link of structural similarities and differences with the proposed prediction, information on the formation of common compounds, or the common underlying mechanisms of the effects of different compounds. I will now give some examples of different types of information from the toolbox which can be used as supporting information for reader cross approaches. I will briefly show how the information should be submitted in Euclid, and then also point out some of the common shortcomings we have noted in reader cross submissions using the toolbox. The following slides show an overview of some rough assessment elements and related to that the type of information that can be generated with or retrieved from the toolbox that can contribute to these assessment elements. For example, the toolbox provides information on the identity of the substances. And for the category description, an important element is the definition of the category boundaries. The structural and mechanistic profilers of the toolbox can contribute to that. In particular, there is more detailed information on the profile alerts available as background information in the toolbox. Another element concerns the impact of impurities. The toolbox can help by predicting properties and modes of action of impurities, or even contains related experimental data. The quality of the source data is very important, as we know. The toolbox includes metadata and references, when available, for the experimental data in its databases. The user can assess the information and also refer back to the original references to evaluate the quality and completeness of the data to be used as source for the reader cross. For the evaluation of the formation of common and non-common compounds, the toolbox can be used to predict transformation products or retrieve actually experimentally observed metabolites. However, quantitative kinetics data are not included. For evaluating the impact and effects of metabolites, common and non-common compounds, profilers, data, and Qsr models in the toolbox can be used. There are also specifically profilers, data, and Qsr models in the toolbox related to degradation and bioaccumulation, which can contribute to assessing these aspects. For considerations on the common mechanism for the similar effects caused by different chemicals, mechanistic and endpoint-specific profilers can be very useful. Available experimental data or predictions can also contribute to reflections on qualitative aspects of the common mechanism. Overall, expert judgment is needed to find the relevant and adequate information that can be used to support the reader cross hypothesis. And now I will show you how to report reader cross in Euclid. It's not sufficient to upload only an automatically generated report from the toolbox. There are specific explanations in the linked guidance on how to prepare registrations. In short, there should be one endpoint study record for each source substance, providing the source data to the reader cross. And these should be completed as any other endpoint study record that documents an experimental study. The study type is normally experimental study. And then there's one endpoint study record for the target data for the registered substance. The study type here is reader cross. And this will be in the same endpoint in Euclid as a source study or source studies. The test material here is the registered target substance. And this target endpoint study record should include the justification for the reader cross. And a cross-reference should be provided as a link to the endpoint study record of the source studies for analog reader cross. There are also some short video tutorials linked in the indicated guidance document and also mentioned on the slide. In terms of providing all necessary supporting information for reader cross submission and a reach, the Qsr Toolbox has some limitations. The experimental data included in the toolbox do not always include the level of detail required for an independent assessment of the study reliability. They do not always correspond to robust study summaries. And the toolbox is very helpful to provide information on potential or observed metabolism. However, details on kinetics, metabolic rates, and the probability of reactions are not included. And these are required for justification of read-across hypothesis. And by missing this supporting information, this can lead to non-compliance of the read-across submissions. But the user can avoid that by adding additional support information from sources outside of the toolbox. Other reasons for non-compliance are related to shortcomings in the use of the toolbox. And this can be avoided by better understanding the functionalities of the toolbox and the requirements of read-across under REACH, and by providing better documentation. It's very important to know that expert judgment is needed for the use of the toolbox. Toolbox is not a black box to produce a ready-to-submit read-across adaptation with just pushing a button. For example, critical assessment of the analogs obtained is needed to check if they are really adequate for the considered read-across approach. And also it's needed to critically evaluate the relevance and reliability of the source data. Common shortcomings in read-across submissions using the toolbox. The whole composition for both source and target substances need to be addressed. As already mentioned, there's the constituents, impurities, and additives where applicable. And sometimes we see wrong or not representative structure is used to run the search for analogs, which then gives obviously not adequate analogs. In many cases, the analogs selected are not sufficiently similar to the target substance. Often not all functional groups of the targets are covered, or the analogs have diverse functional groups and reactivity patterns and therefore cannot be considered as a group. And no justification is provided explaining the possible impact of these structural differences on the read-across prediction. So generally, it's very important to have a clear definition of the applicability domain with relevant inclusion and exclusion criteria that identify the structural requirements and the ranges of physical, chemical, environmental, fate, or toxicological properties for the category. To define the ranges within which reliable predictions can be made for the category members. The user should also always think about whether the definition of the nearest neighbors is based on relevant parameters. There might also be a bias of picking some specific analogs in the category for the read-across prediction without any justification. As mentioned already, it is an issue if not complete data sets in the form of robust study summaries are provided for the source substances. As read-across predictions, we often see a calculated average of no observed adverse effect levels, NOELs, being read across. This is not good because firstly, the most conservative approach should apply. And secondly, in general, not only a NOEL should be read across, but the whole study. Details on the types of toxicity, target organs, and other key parameters are important to judge the hazard of the substances. And often the toolbox is also used for providing supporting information to prove similarity of analogs found outside of the toolbox. To mention again, profilers on their own without bridging studies are not proving toxical similarity sufficiently. To summarize what is needed in a read-across adaptation for REACH, and this applies to all read-across generally, and therefore is also needed for read-across submissions using the Qsr Toolbox. Proper characterization of the source and target substances, a hypothesis showing the relationship between structural similarities and the prediction. It's important to explain why the similarities do not impact the toxicokinetic and dynamic properties. Adequate supporting information for justification, and source study data with good quality. In conclusion, the toolbox with its functionalities and databases is a valuable tool to support read across. And for read across under REACH, the toolbox can be used to identify analogs and the related experimental data to provide supporting information for read across justifications. However, it should be noted that not all necessary information can be covered with the toolbox alone. The toolbox read across is evaluated as any other read across approach, and it needs to fulfill the requirements of an adaptation of the standard information requirements under REACH. Expert judgment is required and critical assessment is important. And to reiterate, adequate justification, documentation, and supporting information is key. Thank you for your attention.
This presentation ends the presentation part of the webinar. Now we move on the Q&A, where our live panel will reply to your questions. Before we introduce the panel, here is a quick recap on how this panel will work. It will run until 6:30 Helsinki time. You can keep sending your questions at slido.com by adding the event code Qsr Toolbox 22 or directly with the QR code, also on this slide. As mentioned before, we will do our best to answer everything we can, but depending on the number of questions, we may not be able to answer all of them. All questions, including the ones we cover in the live panel, will be replied to in a Q&A document that will be available shortly after the event. You will find it on the webinar page, and we will announce it in our weekly news bulletin. You can subscribe to it at echa.europa.eu/subscribe. If you have questions after the webinar, you can reach us through our contact forms from the address on the slide. In Slido, you will also find a short feedback pool. I hope you take the time to share your feedback with us so that we can continue to improve our future events. We will start with the Q&A soon.
Okay, welcome to our live Q&A session of the webinar. My name is Tomasz Sobański, and I'm working in the Computational Assessment Unit at the European Chemicals Agency, and I will be your moderator today. Before the webinar, we have received over 600 registrations and over 30 questions, and we are very thankful for them. Therefore, we did a last-minute changes to the program to cover some of them already in our presentations and demonstrations. Additional questions will be answered during this panel by Doris Hirman and Andrea Gissi. Unfortunately, Andrea Richards couldn't stay with us for this session, therefore some of your questions will be also answered by me. Questions concerning material presented by colleagues from OECD, US EPA and LMC will be forwarded to them, and the answers will be published shortly after this webinar. In addition, I would like to remind you that for any technical questions about the installations of or functionalities of the Qsr Toolbox, you can contact the Qsr Toolbox help desk run by the developers of the tool. You can contact them using the help desk form available at our website www.qsrtoolbox.org. Finally, in few days, we will also publish our answers to your questions in writing, including those that we may not have time to answer today. And after this introduction, let me start with a question that we have received with the pre-registration form of the webinar. Doris, what is the experience and educational background that a user must have to use and interpret the results obtained using Qsr Toolbox?
Hello, thanks for the question. So what experience a user should have? Regarding the use, it should be relatively straightforward to use the simplified user interface or the web client, which we have seen earlier, and to use the basic functionalities of the Qsr Toolbox. If you want to have more options, then the classical user interface is better, but this needs some training and you need to get acquainted with the software. I've shown today some of the functionalities, and I can recommend to look at the tutorials available at the Qsr Toolbox website. They give further instructions, and they are quite a good help to learn more and how to use the different functionalities. Regarding the question what experience or background a user should have for the interpretation of the results, well, it depends. In a simple case, you can use the toolbox to access experimental data only. And then you need to know only how to interpret such experimental results. When it comes to find appropriate analogs or to justify a read across, knowledge on the endpoint and how the structure is related with the effect will be needed. If you know how to do a read across or how to interpret the Qsr predictions, then the Qsr Toolbox will give you a wealth of tools to help you with that. The software has a large knowledge base integrated, and this should help you to make the right choices and good choices within the toolbox. I would just like to note that from my experience, the toolbox itself, it doesn't guide you through step by step, but it rather offers the tools which you can use in various ways. So a certain level of knowledge and some training will be needed. And if you use the toolbox to help you with the REACH registrations, you need to add manual justification in addition to the automatically generated reports. So for regulatory uses, it's also essential to know about the regulatory requirements and also that you are compliant with these.
Okay, thank you very much, Doris. Next, we can answer a set of questions that we grouped together, and they refer to applicability of the Qsr Toolbox to different classes of chemicals, including non-organic compounds, salt, and natural extracts. And the question was, can we use the toolbox for those type of substances? And I think I will take this one. And the short answer is yes, you can apply toolbox to any substance as long as it can be represented by chemical structure, you can find suitable analogs with relevant data, and they are profilers able to capture the specificity of your chemistry. Of course, this task will be much easier for organic substances, as most of the data in the toolbox and toolbox functionality extensively cover those type of substances. But there are also some data and profilers for other type of chemistry. In addition, please remember that toolbox is not really a static tool. Every year, there is more data included in the toolbox covering wider chemistry. Therefore, predictions not possible a few years ago might be possible now. Now, concerning natural extract or substances with complex composition, it is not easy to provide extract structural representation, but you can still try to build predictions for individual constituents or so-called representative substances. To conclude, in principle, the toolbox methodology is not limited to any particular chemistry, but due to the differences in the data availability and coverage of the profilers, there are chemical classes for which will be easier to obtain reliable prediction. And I think that would be the answer for this one. And now, Doris, we have a question about the procedure to follow to identify analogs for read across. Do you have any recommendations for that?
Thanks for the question. For each, you need structural similarity and mechanistic similarity. So you can look for analogs that are similar in structure by using empiric profilers, such as organic functional groups. But then you also have to make sure you have a mechanistic similarity, so you can search for mechanistically relevant analogs by using the mechanistic and endpoint-specific profilers. In the toolbox, you can get highlighted those ones that are relevant if you define the endpoint. Regarding the strategy, if your substance gives any mechanistic alert related with the endpoint, then this alert is the good starting point to look for analogs. For some endpoints like the mutagenicity or skin sensitization, you can also group substances based on the profile of the metabolites. After you have grouped based on the mechanism, then you have to make sure that the analogs are also structurally similar. And this structural similarity, this is really a must for read across under REACH. If you have a substance that doesn't give any alert, then you can start directly with the search for structurally similar substances. And there is also a profiler called structural similarity, which calculates the similarity as a percentage. So if you have too many analogs at the end, you can use that at the very end to reduce basically your group and only keep the most similar substances. So just to repeat, if you want to apply the toolbox, you want to have analogs that are similar regarding the structure, but also similar regarding the mechanistic profile, and you can do that with the Qsr toolbox. To have just one functional group in common, it's for REACH purposes not enough. And also very important, you should make sure that you clarify or discuss structural differences between the analogs for REACH purposes. And at the end, it's also good to check with a critical look if the analogs make sense at all.
Okay, thank you. The next question is about how to submit the model output files. A participant notes that copy and paste of the Word or Excel or PDF into the report for the submission leads to very large reports. And Andrea, can output files be submitted as attachments?
Yes, hi Tomasz and hi everyone. So I think this question is valid for all computational results, not just those that come from the toolbox. And it's very important to make a distinction between Qsr results and read across results. When it comes to QSARs, what we require is to fill in the Euclid endpoint study records, all the fields that are there, include your own justification, and this often requires to go beyond what the tool automatically produces, and then still you can attach the full report as an attachment to Euclid. While when it comes to read across, I think you should just do as indicated in the read across presentation and follow the guidance document and videos linked there. In short, this means that for each source study, you fill in an endpoint study record, and then you, in addition, also prepare one endpoint study record for the justification. And in this endpoint study record, you can also attach the toolbox reports. And I hope this answers.
Yeah, I think it does. Yeah, thank you very much. So let's move to a series of questions that ask about which endpoint or information requirements can be covered by the Qsr toolbox. One is about higher tier endpoints, whether toolbox can be used to predict developmental and reproductive toxicity. And I will try to answer this as well. And in principle, REACH allows the use of adaptations such as QSARs or read across for all standard information requirements, regardless of their complexity. The condition is, however, that an adaptation must provide an equivalent level of information to the standard test. And this requirement in practice differentiates between less complex endpoints like acute toxicity or in vitro genotoxicity and more complex ones like toxicity to reproduction from the question. And for those simpler endpoints, the number of parameters measured by the standard in vivo test are limited and relatively straightforward, like mortality in oral toxicity test expressed as LD50. Therefore, Qsars can produce results with equivalent level of information compared to this test. And basically, they can be developed and applied. In contrary, experimental studies for more complex endpoints measure many parameters related to fertility, reproduction cycle, or offspring. Therefore, they are not currently Qsars able to cover them at the level which is equivalent to the experimental study. Therefore, at the moment, we believe that the best way to minimize the need for animal testing for more complex endpoint is read-across. Qsr Toolbox can be used to support preparation of reliable read-across, and for that, I would encourage you to follow the approach presented today in read-across presentation, and basically as well already covered partially in Andrea's reply to the previous question. And then next question is about regulatory acceptance. Can you please elaborate if there are any changes regarding regulatory acceptability of Qsr results outside supporting evidence and the read-across framework? Andrea, could you take this one?
Yes, thanks, Tomasz. Looking at the question, I think, well, this can be interpreted in two ways, either on whether there have been changes to REACH about acceptability of the adaptations or changes to ECHA policy. Well, I will try to answer both, as we don't know what it was really meant in the question. When it comes to REACH Annex 11, this has been recently updated with some clarification, but the essence has not changed in terms of then regulatory requirements. One thing to note is that recently, with the approval of the OECD guideline for defined approaches for skin sensitization, we start to see a kind of promotion of in silico results that become standards rather than adaptations, and I think this is good for their acceptance. The other way to interpret the question is whether there have been changes to ECHA's policy on the acceptance of adaptations. And I think here the material that we make available evolves, and we try to be more and more precise and transparent in the way we do the assessments. And with this, we hope to facilitate registrants improving their registration and therefore being able to accept more when it comes to evaluation. The latest information on this respect for the Qsars, I will say that the ECHA webinar on the evaluation of Qsars that is still available on our website, we did it last year, is the most up-to-date material. And we have also the practical guide on how to use and report Qsars. For read-across, I think the best is to refer to the read-across assessment framework and all the related material. For the toolbox, this webinar is obviously the most recent we have. And maybe something that is good to know for future, the OECD is running a project called the Qsr Assessment Framework. And this project should deliver two checklists, one to assess the validity of Qsr models and one to assess the validity of Qsr predictions. Now, when this checklist will be ready, and the ECHA is in the lead in one of these work packages, when this checklist will be ready, then obviously you can expect ECHA to reflect the checklist in the evaluation. But we are talking about a year from now, so we need to wait a little bit.
Okay, thank you, Andrea. And now we have a series of questions about the use of Qsr in combination with other evidence, such as in vitro. One question was, can the toolbox be used in combination with a validated in vitro study, for example, acute oral toxicity, to cover an endpoint requirement under Annex 7 of REACH? And I think we've seen today already the combination of in silico and in vitro results applied for defined approaches for skin sensitization, where the final outcome being the combination of different type of tools, so Qsars and in vitro. And this became the standard way to cover information requirements for skin sensitization. Unfortunately, we don't have yet standardized approaches for other endpoints, but already now the additional in vitro information can help building a case to support mechanistic similarity in read-across or as additional evidence to confirm a Qsr prediction. And yeah, and to continue with regulatory acceptance, we have also received a number of questions about the acceptability of Qsr results in different regulations, within and outside ECHA mandates. What we can tell about this, Andrea?
Well, of course, I think we need to distinguish the regulations for which ECHA is responsible and those where someone else does the assessment. I've seen a couple of questions for EFSA or regulations for which EFSA is responsible. So maybe let's start by saying what we see in ECHA, and then let's see what we can tell about the others. We see the use of QSARs in a process called the biocide tier 2 technical equivalence, which means that the impurity toxicity profile of new biocidal products is examined also by looking at QSARs, and then data required only if QSARs show some excess toxicity or excess concern. We also start to see QSARs in proposal for harmonized classification and labeling, but this is very new, so I cannot tell you if this is accepted or not. And then when it comes to regulations for which ECHA is not responsible, well, I would advise to just refer to the specific regulation and authority of competence, because we have heard testimonials of successful applications, but it's not our place to tell what to do and what not to do for those regulations.
Okay, yeah, thank you, Andrea. Another question which I think I can take is, I have a doubts about the application of this software. Is this useful for users or manufacturers? And to answer, from our experience, we know that Toolbox can be used for various applications, and it is used for various applications by different stakeholders. And I give you a few examples. And quite many applications we are aware of are regarding R&D applications. So basically for manufacturers, when they are trying to predict the toxic profile of the new candidate substance to avoid investing too much in product which might not be successfully introduced to the market. So this is one of the quite common use of the Toolbox. Then once you are fine with your product, or once you have your product, then you may need to register it. And then for the substance registrants to cover, the Toolbox can be used to cover also information requirements of this substance. Then quite many regulators use Toolbox mainly to screen or prioritize the substances which would need some regulatory attention. And finally, scientists are using Toolbox to validate their models, or they are using Toolbox as a dissemination platforms for their tools. As you've heard, we have the plugin possibility to the Toolbox, and there is more and more tools every year available as a plugin to the Toolbox, which have been developed by other teams and other researchers. So I think it's quite useful to not for many different stakeholders and for many different applications. And I hope I answered this one, and then let's move to the another one. Andrea, what do you think is the difference between the Qsr Toolbox and, for example, the US tool called ICE?
Well, I would say to answer this question properly, one would have to know a lot about Qsr Toolbox, but also the ICE tool. I don't know so much about the ICE tool. I think ICE stands for the Integrated Chemical Environment and is developed in the US. I've just seen a training that was recently released, and that's all I know about the tool. So not an expert, but my first impression about it, well, first of all, it's a great tool. I recommend you to look it up and maybe see if it helps your purpose. Looking from very far, it looked like it could be repeating what the Toolbox does, but in reality, it seems a tool that is absolutely complementary to what the Toolbox does. First, the focus of ICE is data analysis, comparison of data sets. There is a lot of information about in vitro studies, even those response curves, which goes beyond what Toolbox has for in vitro data. Well, I think for the in vivo side, the Toolbox has more. Two other great functionalities of ICE are those related to IVIVE and PBK, things that are not covered at all in Toolbox. So I think there are very good reasons to also look at this other tool. When it comes to finding analogs, what I understood is that ICE only works on Tanimoto similarity, so it lacks a bit of functionalities of Toolbox to look for mechanistically similar analogs, provide information on mechanisms, and so on. And from what, again, I understood just from training I watched in these days, when it comes to making prediction and data gap filling, it has, okay, the Qsr parameters from Opera, but doesn't go all the way as the Toolbox to really make data gap filling in terms of read across and trend analysis. I will say that, I mean, the good news for all the users is that both tools are for free. The people developing this in the United States are our partners in many projects. And I think the best for everyone is if we will try to get in contact and build synergies among the two tools. But again, if you have a more specific question about ICE, I'm certainly not the person that can help you, but they start to release a lot of material to know more.
Okay, thank you very much, Andrea. I'm looking at time, and I think we have time for one more question. And what about this, Doris? Could you tell us something about the possibility to provide more training on Qsr Toolbox?
Yeah, thanks. About training, I would recommend to go to the Qsr Toolbox website. There is many tutorials available for different functionalities, so please check it out. ECHA plans also to make available more and more material. So also like this webinar here is like a one attempt to give more training. And we also try to include not only the technical functionalities, but also to cover regulatory uses. So please follow us on our communication on the website and also on our social media communication channels. And yeah, stay tuned. There will be more coming.
Thank you. And just to remind you, our Toolbox website is under the address of www.qsrtoolbox.org. And yeah, we are really encourage you to visit this website and see different materials and as well help desk form and get all the updates and news. Okay, so that was our last question in this Q&A session. I hope you got your questions answered. And I would like to thank you all for providing many interesting questions, and special thanks to our panelists for addressing them. Thank you, Doris, and thank you, Andrea. And as stated earlier, in the coming days, we are also going to provide answers in writing to the questions we addressed in this session and to those that we did not have time to cover. And with this, let's move to Tiago Pedrosa's presentation, and he will talk about the future of the Qsr Toolbox, and we'll close this webinar with some concluding remarks. Thank you very much for your attention, and have a great day.
Hello, everyone. My name is Tiago Pedrosa, and I am the Head of Unit of Computational Assessment Unit at ECHA. I hope you have found this webinar informative and useful for your work, and that you had the opportunity to have your questions answered during the question and answer session. It has been exciting to see the evolution of the Qsr Toolbox over the years to provide much more than a Qsr predictions, and how its interoperability is increasing not only with the Euclid, but also with other databases and models, such as Oncologic, becoming a truly global tool. We strongly believe this is the way forward to have reproducible and transparent chemical hazard assessments using non-animal methods. We know the work is not complete, and I would like to take the next couple of minutes going through some of the developments and new functionalities we are already foreseeing in the short term for the toolbox. Having released the last version of Qsr Toolbox last month of March, we are now planning to have the next version released in the first half of 2023. Where we will focus on 3 main areas based on the feedback that we have received from users like yourself, but also on our observations, particularly from the use of Qsr Toolbox in the context of reach. So we will focus the developments on the extension of Euclid searches to endpoint study records. Today, you have seen how to perform structural searches in Euclid data with a dedicated functionality in the Qsr Toolbox, we want to extend the Euclid search functionality so that you can also search data in endpoint study records. We will look into simplifying the selection and application of QSR models. The number of external QSR models included in the toolbox keeps growing, which is great, but it has an impact on the user friendliness and performance of the tool. And the new developments will tackle this issue. And last but not least, we will update the reporting functionalities. Concerning these last points, looking into more detail on the reports updates, as Andrea mentioned in her presentation, the toolbox is a valuable tool, but there are limitations and shortcomings that can impact the regulatory use of its results. Some of these are related with the documentation and justification of the results. In this respect, the report templates are being improved to include link to the regulatory requirements with a completely new justification document, improved reproducibility, as you know, critical for the acceptance of the assessment, and increased transparency so that the underlying information can be assessed even if you do not have the toolbox installed in your computer. We will add the new report in Toolbox to facilitate users in preparing the justification for their read across our QSAR approach. The document guides the users to link the provided information with the read across assessment framework and QSAR regulatory requirements. This information is going to be filled manually, but the templates will include hints on how to better link the information to justify the different assessment elements. Concerning the improved reproducibility, the report will include a new protocol session describing the steps to obtain a prediction. And to increase transparency, the reports will include links to the documentation of used databases and profilers, and will provide more extensive coverage of required information. For example, more information on the identified analogs, including a highlight of the structural differences. Our expectation is that by introducing these changes in the report templates, we highlight elements that have been identified as shortcomings in applying the read across assessment framework. By doing so, REACH registrations and toolbox users in general will be in a better position to prepare and submit better assessments and justifications, and therefore increase the regulatory acceptance of the information submitted. To conclude, I would like to reiterate that ECHA does not only continue to support the development of OECD QSAR Toolbox to promote the use of non-animal methods, but also uses its experience to improve the toolbox to extend the regulatory acceptance of its results, so important to effectively reduce the use of animals. The connection with Euclid, the most widely used international format to share data on intrinsic properties of chemicals, further expands QSR Toolbox functionalities and interoperability, representing an important efficiency gain to all. Finally, it is relevant to mention that the further development of the toolbox is not done in isolation by ECHA and OECD, but also other partners, such as the US, as you have heard before, contribute to its success. And with this, we arrive to the end of this webinar. I hope you have found this webinar useful. And on behalf of me and my colleagues, I would like to thank you for your participation. Thank you and have a good day.